Well, we'll go ahead and get started with our final oral presentation. It's my pleasure to introduce Dr. Anna Cohen. I had prepared notes of what her roles were, but I see they're right here, so you guys can read them as well as I can say them. But she has both clinical and research responsibilities. Clinical as Assistant Director of Clinical Molecular genetics, and also from a research perspective, her home is in the Center for Genomic Medicine here. Dr. Cohen is a recipient of a pilot award from Frontiers. It's actually quite interesting and has spawned an R01 submission that should be going to study section this month. Yeah, so hopefully we'll get good news. I think it's a pretty cool story, but we'll let her tell it. And just so there's no pressure on Dr. Cohen, she's the only thing between you and the reception and heavy hors d'oeuvres right after her.
No, she's not.
There's closing.
Well, there's closing remarks, but we're not going to be the rate limiting step.
Well, thank you, Steve, for that introduction, and I hope I'm just gonna, open your appetites for celebration after my presentation. So it is my pleasure to be here today to talk to you a little bit about particularly the project that was gracefully funded by Frontiers by the Pilot Award, and I'm really grateful for that because we learned a lot. So here we go. Just for some background, I want to touch upon the state of rare diseases in genomics. So there are over 7,000 rare diseases. They're individually rare, but collectively common, and really account for about 1 in 6 children's hospital admissions with a very high death rate in early years. Unfortunately, a lot of these families, and particularly kids, go through a very long, what we call a diagnostic odyssey, which is a long time to get to a diagnosis and understand why they have these genetic disorders. The average, which is estimated around 5 years, also goes through a lot of misdiagnosis and misunderstanding from providers. And currently, the diagnostic rate is about 25 to, we're getting up to like 30, 35% clinically. And even though a lot of these disorders don't have necessarily clear treatments or definitely not anything that's going to solve most of their problems, it really helps determine how to manage their care better, to definitely avoid interventions that are unnecessary. But most importantly, a lot of the families really appreciate having a connection with other families that might be going through the same thing and just having answers. So what are the challenges that remain? I mentioned there's about 25 to 35% of families that get a diagnosis. What are the challenges that remain? Why is it not 100% or even close to that? So one of them is knowledge. We don't have knowledge of all the causes of genetic disorders. Could there be more things happening? Is it not just one gene that's causing one disorder? Could it be multiple genes? Could it be genes and environment? Could it be just an exposure in utero, for example? Could it be also a big thing is technology limitations? Are we using a technology that's not picking up a genetic abnormality based on just that technology not being able to see that? But one that is more what I'll focus on today is a lack of access. So is genetic testing even utilized and accessible for all of these families? So quickly touch upon my research home here at CMRI, which is the Genomic Answers for Kids initiative. It's really where a lot of these efforts to advance all of these challenges is housed under the leadership of Dr. Tomi Pasanen. You can Google it and find out more information. But really, I'd like to highlight, first of all, that it's truly an unparalleled genomic initiative in this hospital, and it really focuses on a strong collaboration between the clinical and the research sides. It's such a strong partnership that we've now been able to help increase that diagnostic rate from the clinical side, both with just reanalysis of data and the addition of new technology. And we were able, on the clinical side, to launch long-read genome sequencing here, which no other institution is doing. So it's pretty exciting. And obviously, we're generating a lot of data, and all of this data is shared in a de-identified way so that we can advance the field and not just our own research. But importantly, I like to quickly highlight that Genomic Cancers for Kids is more than just this diagnostic machine. There's a lot going on. We're also developing these new technologies and analysis tools. We're also collecting other samples that allow for functional studies. For example, we have a group upstairs that does cardiac organoids and brain organoids. It's super cool work, and you get to test therapeutics on those. There's also work going into more complex disorders and oncology, so it's truly a multifaceted program. But let's get back to the main topic of today, which is the limitations in access to testing and what I've been focusing a little bit more on my research time. So there's two primary barriers in access to testing. One of them is financial. And that will include, obviously, in the United States health system, insurance denials. So a lot of kids go through, medical assessment, and it's determined that they should get clinical testing for a genetic disorder, but that might be denied. And a lot of these families do not have the resources to pursue the testing. So this is unfortunate, but it's still very much of a problem. And then the other barrier is access to these providers and to this testing. How can the families even get there? So here's a map that shows Kansas and Missouri, which I believe everybody in this audience would be familiar with and able to recognize. And this was work done to look at the enrollment of the Genomic Cancers for Kids study and where that enrollment was distributed. And you can see, so just think darker, so the red, heavier enrollment down to gray, very light enrollment. And you can see in the center is where we are located, and the further you get away from our center, the least enrollment there is. And this is truly very heavily focused in Kansas City and surrounding, and basically a large urban center. All of this data was pulled by Dr. Mark Hoffman's group. It also shows racial and ethnic inequalities as well as socioeconomical inequalities. And in fact, when you zoom in to Kansas City proper, you actually notice that there's less enrollment in neighborhoods that are closer by, but socioeconomically disadvantaged compared to further away suburban areas that are more advantaged. And that is definitely not what we want to see. We want everybody to have the same access to testing. And in turn, this also contributes to a lack of diversity in genetic data, both within our system and in the wider rare disease community. So definitely there's limitation in access to testing, but there's another issue that providers keep mentioning, which is there are too many patients that are waiting to see genetics to even get the testing order. The genetic services are completely overwhelmed with referrals, and it's really hard to get to see them to even begin the testing. Wait list is around 6 to 12 months, and then after that, if insurance approves it and they get the samples and there's no issues, it still can take a while to get to an answer. So we estimated, based on internal data, and diagnostic rate of approximately 8% at 16 months, because a lot of kids are even coming in for reasons that they shouldn't come in. So it's really a low yield here. So what can we do? And this is really where the pilot project focused, is we wanted to reach these underserved populations around us. And we decided to use our resources here to support providers that are in local communities and can access their patients at their local clinics. So instead of asking the families to come here, we're coming to them and providing all of the sequencing and all the support. And really that requires a lot of empowerment of primary care providers to feel comfortable nominating their patients to the study, and also understanding what that even means for follow-up care. There were a lot of lessons that I look forward to sharing with you. This we envision not just as a solution here, but it's something that could be transferable nationally because there is a shortage of genetic specialists in the entire country. So instead of coming to see genetics and then ordering tests and then having some diagnosis, what if we flipped it around? So this is really the vision. Instead of having a subset of patients who make it to genetics get testing and a subset of those have a diagnosis, let's test everybody that might have a diagnosis, the ones that either have a clear positive or a completely negative result, then go see genetic specialists. And then that should reduce the wait list for everybody really seeing genetics. And you should end up with a subset of patients that actually need referrals. So we're flipping around the axis here. And all the testing, obviously, here for the pilot was covered by our research study. But because we have the strong partnership with our clinical lab, all the results from the research study get confirmed clinically and given back to patients in a clinical report. So as I mentioned, and you've heard, our pilot study was funded by Frontiers, and here's the results. We had 30 nominations. Turns out a couple of them had already been enrolled previously through a different provider, and some we were unable to complete consent just because it's hard to reach them. We partnered with this clinic in Salina, Kansas. I got to go there and meet them, and I saw just quite how isolated it is. But we had very engaged providers and that made this study really successful. So we ended up at the end with 23 families enrolled, for which we got samples for 21. And of those, we actually got 5 diagnoses, which is pretty high. And we were quite surprised. That's about almost a 24% diagnostic rate. And for 3 others, we actually have really strong candidates. And if we were able to either get samples from additional family members or pursue functional studies, which we are doing, we would actually raise that diagnostic rate to like 38%, which is highly impressive. And then one thing that was quite surprising to me personally was that 3 out of the 5 confirmed diagnoses were actually large copy number variants. I'm not going to go into details of all of these, but I also wanted to point out that 3 out of these 5 are also kids that were aged 7 to 16, which is older than what you would typically expect for an initial genetic diagnosis for something that's we would call kind of an obvious diagnosis. It was an easy to find answer once they got the appropriate tests. So really, the main outcome of the study is the diagnostic rate was higher than historical external referrals to CMH. We think that's because these kiddos had virtually no testing done previously, and also our relationship with the providers to really recognize which kids should be getting tested. But the most important thing is so much faster than the traditional refer to CMH, see a specialist, order the tests. All of these were reported under 4 months, and just the wait list would have been 6 to 12 months. So it's truly revolutionary, and also the patients did not have to come here. We came to them, and just as I said, some of these were older than we expected. So we're kind of wondering if there's this age group that if they're not picked up in early intervention years, that they're just going to go to school and possibly struggle at school and not have support and never really pursue genetic testing. There was a lot of lessons learned as well in terms of like how hands-on something like this needs to be when you're collaborating externally. The biggest challenge that I really want to highlight, because this was a big lesson for me, is that we anticipated that having that result in hand would empower families to then get a lot of services and support, for example, like special support at school for their kids, and turns out that's not how the system's built. So there's larger systemic problems really out there that are not allowing these families to access resources, even if there is a genetic diagnosis in hand, and that was kind of heartbreaking and very informative for next stages of educating providers that having a diagnosis is always going to be helpful in terms of having an answer, family planning, and managing their symptoms, but it might not actually change their lives day to day as much as I had hoped. So it's very humbling as well. So quickly, what's next? Truly, a lot of lessons learned, a lot of troubleshooting, and we're expanding. So we've now established partnership with an additional 5 clinics in rural Kansas, 4 in rural Kansas, 1 in rural Missouri, and we just received the first, actually we just received a second family from this new partnership, so we're really excited. And following the feedback of the pilot, we also found some community organizations that are out there and closer to these clinics that can help us with the connection between the families and local resources, which is something that's hard to do from far away. So conclusions, by providing local support to the patients at their local clinics, we are bypassing the bottlenecks, the delays in getting to genetics, and we're really trying to integrate these genetic diagnoses into primary care at local rural clinics. We really think this also represents one step in improving equity and access to rare disease testing, but also research and personalized care. And we really, like I said, this is very promising, but remains with a lot of challenges, especially for the families that may not actually have access to better care after. But we are still pursuing forward with just improving access to genomics. So with that, I am going to conclude with saying thank you to all collaborators of Genomic Answers for Kids. Thank you to the families that participate in our study, all our funding, and obviously Frontiers for supporting my pilot project. And happy to take questions.
Thank you very much. While I walk, okay. I just wanted to say, one of the things that this current cycle of funding for Frontiers focuses on, two of the themes were rare and rural, and this project kind of hits both. And just as a heads up for those of you who are interested in what Frontiers has to offer, one of the things that we are now pursuing is a practice-based research network. Dr. Castro is leading that, so stay tuned. Advances in healthcare that come out of Frontiers and its investigators should not be limited to those who live within a short distance of a tertiary care academic center. And we very much would like to be able to support initiatives that take what we have to offer outside the confines of the Kansas City area. So with that, Dr. Monahan Nichols.
Yes, that was a wonderful talk. It's a fabulous project, and I have a couple of questions. So for the patients that had unidentified, you had 13, I think, or something like that, patients that had unidentified alterations. Was that compared to family members that didn't have the same condition, or could there be unidentified? Could there be alterations there that you cannot conclude what they are? Not necessarily a variant of unknown significance, because it's pretty clear that the 3 that you have an idea that it might be significant. But what was the implications of the unidentified? Was there nothing found in their genome, or did it not work, or was it enough? So what was the?
Okay. To clarify, my understanding of your question is what is happening with the families that remain without an answer?
Yes.
Okay.
Yeah.
So when I say there's no answer, I didn't, at this time, with the current knowledge, have any finding in their genome that was of clear clinical significance. Obviously, everybody has a lot of genomic changes. A lot of them will be benign. They just make us unique and different from each other. So really, we need enough knowledge in the field to understand which ones are causing disease. And for those families, we didn't find anything that, based on current knowledge, is causing disease. However, some of them did have, potentially a change in the gene that was like, potentially with more knowledge, one day this could become diagnostic. All of these, even though this was a pilot project, it's under the umbrella of the Genomic Cancers for Kids program, so they remain in the Genomic Cancers for Kids pipeline, and we keep seeking more knowledge. So it's not like it's done, we found nothing, we're never looking at you again. That's not happening.
And of the 2 patients that had chromosomal deletion syndromes, were they novel, or had they been identified and associated with other, seen in other patients with similar symptoms or subsets of symptoms?
One of them had been reported before. The other was just so large that it was clearly pathogenic. That exact one hadn't been seen, but other overlapping ones have been identified. We were very surprised that I would find such a large chromosomal abnormality in a kid that wasn't that young anymore, and that they were supposedly doing not terribly, just needed extra help at school. So,
thank you.
Did the diagnostic pipeline that you use include the methylation data?
So that is a great question. Thank you, Steve. This pilot was done not on long-read sequencing. This was done with exome sequencing only. So it is just looking at, typical exome, but we did copy number analysis on the exome backbone, which we also do here clinically. So it is still limited. So all the other undiagnosed patients are now eligible for long reads. However, we would need to recollect because all of these were done with saliva samples. So when you're working with remote clinics, it's a lot easier to collect, a saliva sample and have that mailed in than having blood drawn and have that sent in. And currently, we're only doing long-read sequencing on blood. But we're hoping to eventually develop that on saliva so that it can reach a lot more families out there.
And so that's part of the answer to Dr. Nichols's question. Don't you have a couple of 1 or 2 cases where there's a causal variant on one chromosome and a completely silenced copy of the other? I thought I saw the poster up on the 3rd floor.
No. I mean, no, that's not the case at this point. I mean,
you don't have the information
because I wouldn't have.
You don't have the long read data. Any other questions?
If anyone knows any rural clinics that might be interested, please do send them our way. We're always looking to expand and work with them. And we've also learned, now that we've made new partnerships, that every single clinic is going to need support in a very different way. And that is a fascinating process as well. So.
Oh, one more.
Well, thank you very much for a very interesting presentation. I am Baki Agbas from Kansas City University. How feasible to test the genomic of the newborn kids before they leave the hospital? Is it feasible? Is that doable? The catching these things, the earlier will provide the clinician not to miss the window of, the treatment window of opportunities. How feasible is this?
Absolutely. So there's actually a lot of efforts out there for looking at doing genomic testing in newborns right away. I think the difference is whether, I'm not sure if you're asking testing every single newborn or just the ones that are sick.
Just when the baby is born, they just get the blood from the,
right.
So heel and then,
right. So general testing every single newborn, we don't believe is, or at least this, my personal opinion is that it's not super helpful because a lot of the analysis that we're doing is based on, we know they have this problem, we're looking for a cause of this problem. Without that guidance of what the health problem is, it's really hard to interpret all the variation that we're seeing, and we're seeing a lot of variation in everybody. So it would be, a lot of the reports would end up negative because there wouldn't be anything that's glaring obvious to give back to these families. However, on the flip side, if we thought about any baby that is born with any complication, testing those, I think that could be helpful. And that's sort of kind of in parallel with what CMH is now trying with the long reads in very severe NICU patients.
Last question.
I was just curious what the prospect is for transferring this research to other aspects of the medical industrial complex, because there seems like lots of opportunities to better serve people by rather than having the specialist need a referral and then you go there because it's like, oh, you broke your ankle, maybe your bones are weak, let's send you to a specialist. You wait a few weeks and they're going to have you a DEXA scan. Oh, well, you're here, you have a DEXA scan, and then come back. Well, I just wasted half a day off work and a couple $100 for you to tell me what I already knew I was going to need. So it seems like there's lots of opportunity for this change in thinking to better serve people to be expanded. What are your thoughts on that?
Yes, that's a great question because I talked a lot about genetic specialists, right? And the kids that go to genetics and then would get testing. So we've actually, through Genomic Answers for Kids, been able to learn a lot because the partnership within Children's Mercy goes through all specialties. So we have nominations from every single specialty in the hospital. And what we've learned is what is the yield of success in finding a diagnosis based on who is referring, which subspecialty is referring. And it is much, much higher for genetics and neurology compared to any other specialty, which is why efforts have been still focused on these for something like this, like an expansion. However, that is not to be said that we wouldn't find diagnosis for other subspecialties. You just have to start somewhere. So we're starting with the highest yield. Obviously, if this ever became, a test that everybody could easily order and that would be cheap and accessible, definitely there is space to order it for a lot more types of medical presentations.
Well, thank you very much for a wonderful presentation and closing out our Frontiers Research Symposium.